Hello guys, welcome back to the master class. So we are continuing our learning of the important essay type questions in applied microbiology for university exam. So in this series, the next question is describe the structure, growth and nutrition of bacteria. Remember, in this channel, the entire explanation will be in very simple English so that students from across India can easily understand it. And one more suggestion I would like to give here. Guys, if you are watching this video on a mobile phone, so please keep the orientation of your mobile phone to horizontal so that you can watch the video in full screen. There will be no distraction. So once you understand the whole concept of the videos here, then mute your audio, then start writing the points. And regarding this particular horizontal position of your mobile phone, this is not only for this channel. For that matter, when you are watching any kind of educational videos, so try to watch that in a full screen when you are using mobile phone, because that will prevent the distraction. Because when you use that vertical position below the video, something else also comes which may divert your mind. So in that case, I would strongly recommend you guys, while using mobile phones, watch it in a full screen. So come back to our lesson. So the question is, describe the structure, growth and nutrition of bacteria. So as I said earlier, for the essay type questions, you have to start the answer with introduction. So in this case, write the introduction like bacteria are microscopic unicellular prokaryotic organisms that lack a true nucleus and membrane bound organelles. You know what are the membrane bound organelles? Because they remain in a particular compartment and perform their individual functions. But that is not there in case of bacteria. They are widely distributed in nature and play important roles in health, disease, and environmental processes. So let's start with the structure of bacteria. A typical bacterial cell consists of the following structure. In this case, you have to draw a neat and labeled diagram. Already you are aware of the diagram. So once again, the capsule, the outermost layer in some cases, cell wall, plasma membrane, nucleoid, plasmid, flagellum, singular, plural is flagella, cytoplasm, pilus, singular, plural, pili, and ribosomes. In this way, first you need to draw a neat and labeled diagram. So guys, after diagram, you need to highlight and describe each and every component of this. Guys, let me explain one more thing. The essay type question that we are discussing now has three different parts like structure, growth and nutrition, and each individual part is also important for short note. For example, in this case, only structure of bacteria is also an expected short note that carries 5 marks. So that also you make a note of it. So cell wall, rigid, and make the bullet points that I told you earlier. Rigid outer covering gives shape and protection. Underline, composed of peptidoglycan, prevents osmotic lysis, and this is very important again. Basis for classification, this cell wall. Gram-positive bacteria, they have a thick peptidoglycan layer. Underline this, MCQ. Gram-negative bacteria, thin peptidoglycan layer with outer membrane. Next, cell membrane, also called plasma membrane, made of phospholipids and proteins, functions, selective transport of substances, and respiration and energy production. Underline this, respiration and energy production is done by cell membrane, biosynthesis of cell components. Next, cytoplasm, semi-fluid matrix containing water, enzymes, proteins, and nutrients. Important here is cytoplasm is the site of metabolic activities. Underline for MCQ. Next, nucleoid. You already know that bacteria do not have true nucleus. So nucleoid contains a single circular double-stranded DNA not enclosed by nuclear membrane. Ribosomes. Bacteria contain 70 S ribosomes present in cytoplasm. Underline this, site of protein synthesis. MCQ. Ribosomes are site of protein synthesis. Capsule, outer gelatinous layer in some bacteria. Gelatinous, it's a kind of protein. Function of capsule. Again underline, protection from phagocytosis, and the capsule increases virulence, means chances of infection. For example, Streptococcus pneumoniae and Klebsiella pneumoniae. Next, flagella, long filamentous appendages responsible for motility, movement. And next is pili, the plural of pilus, also known as fimbriae, short hair-like projections, and they help in adhesion to stick and conjugation, transfer of genetic materials. So this is all about the structure of bacteria. So next, growth of bacteria. Make a note of it. This is also an expected short note. Growth of bacteria with 5 marks. Definition. Growth refers to an increase in the number of bacterial cells by binary fission. This binary fission involving the following stages: DNA replication, division, chromosome segregation, and formation of septum, means dividing wall, and finally division into 2 identical daughter cells. So these are the 4 different stages. So guys, growth curve for bacteria, this is very, very important. You need to draw a neat graph for this growth curve of bacteria. When bacteria are cultured in a closed system, means closed vessels with a limited amount of nutrient, so they show 4 different phases of growth. Let us understand this. This graph shows in the x-axis, this is time, and y-axis, this is log of numbers of bacteria. So it starts with the lag phase, no growth. This is called lag phase. This is very, very crucial. This is called exponential phase. Bacteria start growing in a rapid manner. This is called exponential phase. Then comes the third phase, stationary, means constant. growth remains constant. Thereafter, this is called decline or death phase. Bacteria number of bacteria starts reducing, that is due to the death of bacteria. So this particular graph is very important for you to draw in growth curve of bacteria. Then you need to highlight all these 4 pages. The first one is lag phase. No increase in cell numbers. Bacteria adapt to new environment, and in this particular phase, lag phase, there is active synthesis of enzymes and proteins. This synthesis takes place in lag phase. Then comes log phase. This is very important so far the growth is concerned, also known as exponential phase. In this phase, rapid multiplication of bacterial cells takes place, and this phase is most metabolically active phase. Next, stationary phase, growth rate equals death rate. Nutrients become depleted. Nutrients gradually, the amount decreases, and waste products accumulation happens in this stationary phase. And then comes the final decline or death phase. In this phase, number of dying cells exceeds new cells, means death is more than the new. Population, bacterial population decreases gradually. So these are the 4 phases of bacterial growth. At the end, you need to mention factors affecting bacterial growth: temperature, pH, moisture, oxygen, nutrient, light, and osmotic pressure. So the third part of our essay type question is nutrition of bacteria. Note that this particular section alone is also an expected short note of 5 marks. Nutrition of bacteria. So let us start with the definition. Nutrition is the process by which bacteria obtain energy and nutrients necessary for growth, metabolism, and reproduction. Bacteria require following nutrients: water, essential for metabolic activities, and serves as medium for transport of nutrients and waste products. Underline this MCQ. After water, carbon. Carbon is the main component of bacterial cell material. Underline this. And obtained from carbon dioxide or organic compounds. So next in nutrition, nitrogen required for synthesis of proteins, nucleic acids, and enzymes. And the sources of nitrogen are ammonia, nitrates, and organic nitrogen compounds. Next, sulfur, essential for the formation of amino acids and vitamins. Next, phosphorus, important component of nucleic acid, ATP, and cell membranes. Minerals, calcium, magnesium, potassium, iron, manganese, zinc, copper. These are required in small quantities for enzyme activity and cellular functions. And lastly, growth factors, vitamins, amino acids, purines, and pyrimidines. So guys, if you like the videos, please subscribe to the channel, share with others. My name is Deepak. Stay tuned for the next video. Till then, take very good care of yourself.